Good morning or good afternoon. Hey guys, and welcome to the final FRQ preparation for your exam. This is not so much about going through this particular FRQ as we've already covered it in our practice sessions, but this is basically going to be a short video talking to you guys about the essentials of solving FRQs. So basically, what are the things you need to look for and what information is important? What are strategies that you can use to help tackle these FRQs as you go into the exam? Now, the great thing and the important thing to remember about FRQs is that a lot of it is not content knowledge. Yes, content knowledge is important. And no, I'm not saying that everything I've taught you this entire year should just throw out the window. No, keep that stuff in your mind. But know that a lot of the processes and strategies that you'll be using to solve FRQs are really just boiling down the scientific method to its essentials. It's knowing what's important and what to pay attention to. And so that's really what I want us to focus on today and to take away from this FRQ practice here. Let's go ahead and begin. Now, every FRQ that you're going to see, or most FRQs you're going to see, are going to have some sort of introductory text. And the text is going to describe for you the context situation that the FRQ is based on. It's also going to give you some key details that will be important in answering the sections of the questions that come afterwards. You will also usually have some sort of figure or graph that you will have to interpret in some sort of way, and that figure or graph will also often be linked to the context that you've been given, and you'll have to use it in order to solve some sort of problem. You will then be given either 4 or more parts to be able to break down the information that you're given in the introduction in the graph into some form of answer or analysis. Usually, not always, but usually, part A of the problem is going to be rooted in the content knowledge to talk generally about whatever it is that you're looking at. Part B is going to be more contextual in terms of the graph, so it's going to be asking you to go in and figure out, can you analyze the graph or the figure? Can you read it? Can you draw conclusions based off of data? Part C is usually going to be building off of Part B and having you guys to justify or predict or explain whatever it was that you talked about in B. And then D is usually the application. So D is going to give you some sort of scenario or extend whatever it is that you're trying to look at to apply it to something else. So how should you go about attacking an FRQ? I'm going to start here by underlying some key things within the introduction, because I know the introduction is not only going to give me the context, but it's going to give me some key clues as to how to solve this. So let's go ahead and begin. The common bedbug, Cimex lecturarius or something, is a species of insect that is becoming increasingly resistant to insecticides. Okay, so out of all of this, the common bedbug, and becoming increasingly resistant to insecticides, I'm going to underline becoming increasingly resistant to insecticides, because the name of the thing maybe is important, and you could underline that. But what I got out of that sentence is that we're talking about resistance to insecticides. Oftentimes in their introductions, they will tip you off as to what the sort of focus is. And if you remember something from your modules or your units, you can kind of pinpoint generally the area or the unit or topic that they're going to cover. Here, I'm thinking we're going to talk about insecticide resistance. And so that's sort of where my brain is going to go and how I'm going to interpret this problem. And here we go. Yeah, so bedbugs possess several genes suspected of contributing to the resistance, including, and I'm going to actually underline these because these are the specific names of the things I'm going to be working with in this problem. So P450, ABC8, and CPS. To investigate the role of these genes in insecticide resistance, researchers deleted 1 or more of these genes in different strains of bedbugs, as indicated in Figure 1, and treated the strains with the insecticide beta-cyflorin. Now, just to make sure that I understand what that is when I see it later, because it's a big word that I might not recognize, I'm going to go ahead and underline both insecticide and beta-cyflorin so I can remember what it is. We also get some important information here before I move on. So they went ahead and they deleted 1 or more of these genes. Okay, so basically what they're doing here is they're cutting off the genes in sequence to see which one is most important in contributing to this strain of insecticide resistance. Each strain was genetically identical for the deleted genes. Aha, okay, so when you see the word identical, or they start talking about something being the same, they're trying to tip you off to the control of the experiment. So each strain was genetically identical, okay, except for the deleted genes, and was equally fit in the absence of beta-syflethrin. So that is our control. That is the justification for the comparison that we'll make between the control and the experimental group later to show that whether or not there was an actual difference in the experimental group versus the control. The percent survival of each strain following beta-syflethrin treatment is shown in figure 1. Now, before we move to the figure, these are some of the things that I would recommend doing with this. 1, when they present this information to you, they're going to present it to you in a very technical language, okay, in the language of science, of biology and AP. One of the best things that you can do is to sort of translate this into your own words to make it more accessible. And one of the best ways to do that is to try to take what you just read and to phrase it into a scientific question. Now we know that in the scientific method, all scientists start an experiment with a question. The question is going to really define their experimental setup and how they go about tackling and testing to see whether or not the thing that they want to know about is true or not. So let's see if we can come up with a scientific question that they're trying to get at here when we're looking at this. Now they're talking about resistance to a pesticide, or rather to an insecticide, and they're talking about the genes that confer those resistance. And the key to it is their experimental setup here that they described. They deleted one or more of these genes. If they're deleting one or more of the genes, well, why would they do that? They're breaking the genes one at a time to see which one confers the best insecticide or pesticide resistance. So if I was to come up with a question for them that they're using to do this experiment, I would say, which genes are most important in providing insecticide resistance to bedbugs? Okay, this is a very simple one-sentence question that I'm able to come up with that basically encapsulates the spirit or the drive of this study. So that gives me a much better focus and idea when I'm going into the graph so that it can really help me to figure out what the important details are. The other thing that I might recommend doing here is coming up with the variables if you can. Now the variables actually I think we can come up with here. So we're going to come up with the independent variable here, which of course I'm going to call the IV. And the independent variable here is what the scientists are changing. Now it says that they're deleting one or more of these genes. So I'm going to go ahead and say the deleted genes are going to be the independent variable they're working with. And then the dependent variable, which is called the DV of course, is going to be what changes based off of the independent variable. So I'm guessing if we're talking about insecticide resistance and they're trying to see which gene is going to provide the best insecticide resistance, we'll be looking at the insecticide resistance in some form or fashion, whether it improves or decreases based off of which genes that we delete. We also are given our control, so I'm going to go ahead and put that down as well. In these little notes, now I'm taking them on this program, but a really great program that I like to use oftentimes when I'm working with my computer is Notepad. Just type it into your start and you can bring it up and you can go ahead and start typing the stuff in here. I'm going to go ahead and keep doing it the way I am, but you can do it there and you can save it for later if it helps you. So what is their control? So their control is that the strains are genetically identical except for the deleted genes and equally fit in absence of insecticide. Okay, so I've basically outlined their entire experiment. Why is this important? Because to understand the results of an experiment, you really have to understand how they put the experiment together. If you take these key elements of the experiment and you're able to pull them apart, identify them, and understand them, you can typically understand how the scientists got their results, and you can understand what the results actually mean. So let's go ahead and just keep this information in our minds and move on to the next stage. Now notice, I haven't looked at the questions yet. I don't care what they're going to ask me. This is just something that I went in and I did naturally without even going on to the next stage. Let's take a look at the figure. So the figure is going to show us graphically the data that is supporting their question, whether or not it is supporting their hypothesis or not. So let's take a look at their caption here. Percent survival of 5 strains of bedbugs treated with beta-cephalorin A. A plus indicates the gene is present, a minus indicates the gene is deleted, and error bars represent the 95 confidence interval. Okay, so what's interesting here about the setup of this graph is that the x-axis, or our independent variable, which is which genes are cut on or off, which we can see, okay, we nailed our independent variable here. So that x-axis is in the form of a chart because they tried different combinations of cutting the genes on, or cutting one off at a time, or cutting several off in tandem to see the results. We see that their DV, they're measuring in terms of the percent survival. Okay, so in other words, the higher the percent survival, the better the insecticide resistance, which we can see insecticide resistance was our dependent variable from before. Good, we can understand that. Now, before we move on, we need to understand error bars. Error bars, the standard error of the mean, are basically there to represent how well that data is actually representing the population mean. What I mean by that is, whenever you are doing an experiment, you're obviously using samples. You can't usually use an entire population of individuals for a study because the population is probably just going to be unrealistically huge. How would you ever be able to, individually study each one of those members or take data on an entire population? It'd just be too wieldy. So you use samples that are supposed to be representative of the population. So this data is representing some samples from the bedbug population. Not the entire bedbug population in the world, because that would be impossible, right? You have to track down every single one of those little things. If you did that, you might as well just get rid of them. No, I'm just kidding. They have a function, they have a purpose. So in this case, we're looking at samples. Each one of these bars is, of course, representing data values, and we can take the mean, right, of each of the data values that we're seeing here. And that's what we're seeing. So we're seeing the sample means being represented. The error bars are showing us whether or not these sample means are good indicators that our samples are representative of the population. In other words, that they're showing that this trend is likely within the overall population of bedbugs. Or if the error bar is massive, if it's huge, it's showing us that maybe our sample data really isn't representative of that bedbug population and that we shouldn't have that much faith in our data. The 95% confidence interval is just telling us how confident we are and what metric that we're using. There are different confidence intervals that you can use, 97%, 99%, or less than 95. Typically, though, scientists will always use either a 95% confidence interval or above. This basically means that the scientist says, I am not going to accept this data unless I am 95% confident or better that it is not due to random error or chance. Basically, they're trying to make sure that the thing that they're seeing is an actual pattern and a phenomenon based off of some kind of cause in nature. So when we take a look at error bars, the bigger they are, it basically means that this bar that we see here could either be up here or it could be down here, and that there's basically an equal chance that it's here, here, or here. Okay, so we don't really know. So there's fluctuation. There's a lot of error in this bar. As compared to this one right here, well, I guess I should use this one right here, which has a much smaller error bar in comparison to the size of the actual bar of data that represents the data. So this one could either be this high at 80 or it could be this low at 60, but right now it's hanging around at around 70, and so that sort of gives us an indicator. Now, if error bars overlap, as we can see they do, so in this error bar right here, this error bar, it barely overlaps, but it touches right there. This one definitely overlaps with this one over here. If the error bars overlap, that means that there's really no way to tell whether there's a significant difference between this data and this data. They could be, this guy could be this low, and this guy could be this high, and they might be the same height. This one could actually be higher than this one. We just can't really tell because there's fluctuation here. Okay, this error bar means that this bar could move and be different heights. So that means that there's no significant difference, as surprising as it might seem, between this bar and this bar, which might seem absurd because they look so different just based off of this, right? But remember, the error bars are showing you the potential fluctuation. So there's no significant difference between these three guys right here. And this one, it's a little bit hard to tell whether it intercepts. I think it just barely doesn't. So maybe there is a significant difference between this guy and this guy. There certainly is between this guy and this guy because the bars do not overlap, and the same here and the same here. So we can tell which one is a significant result and which one is data that we might not be able to confirm and say, yeah, this is a pattern that we see. So taking that into consideration, let's just go ahead and read through and see what patterns that we can draw from this graph. Okay, I see here, I have a strain P450, ABC8, and CPS. When all of them were normal, in other words, they weren't deleted, this is how much insecticide resistance we got. It's a lot. It's pretty big. Okay. Now it does overlap with this guy over here, so it means that maybe there's not a significant difference between here. We'll talk about that. But there is a lot more insecticide resistance when all three are left as they are than there are when we delete some. So let's go ahead and see what happens when we delete P450. When we delete P450, it does cause a significant drop. We see the error bars do not overlap with when they were all together. And it's caused a significant drop in survival and thus in insecticide resistance. We see another drop in ABC8, and we see the drop in ABC8, the drop in ABC8 is more than this one. However, the error bars do overlap, so maybe we can't say that there's a significant drop between these two, but it certainly drops a lot farther away from this guy. And then over here, CPS, when we delete that one, we see that the error bars definitely overlap here. So maybe the deletion of CPS doesn't cause a significant enough change for us to say that that gene is very important in its insecticide resistance. It might be hard to tell. When we delete both P450 and ABC8 at the same time, we can see that insecticide resistance decreases a ton. Very few of these bed bugs are surviving after we delete those two. Okay, so I'm going to go ahead and write down those trends very quickly to myself, either on my notepad or off to the side. And the reason I'm doing this is to make sure that I kind of keep all these relationships clear in my head. The whole point, again, is to go ahead and isolate these simple ideas so that we can kind of keep them in our minds as we go forward. Okay, so what am I going to take note of here? Well, I think that from what I can see, ABC8 is the most important gene to insecticide resistance. Okay, I see that one falling the most in terms of its error bar relationship to this guy. Okay, so it's the single most important gene to insecticide resistance that I could see. Okay, CPS is the least important single gene for resistance. I'm just going to go ahead and simplify it for myself. Okay, and that when P450 and ABC8 together, I guess, allow the fewest survivors or something like that. So together, they're really bad. The only significant differences that I see here in terms of the error bars and stuff is we do see that both ABC, well, I should say both P450 and ABC8 cause significant drops in survival. Okay, so both of them, there is a significant difference between them and all the wild types. So yeah, I think that'll do it. So these are all the relationships that I kind of want to keep in my head, but ABC8 is the most important gene, CPS is the least important, and all of this. So let's go ahead and move on to the actual questions. So bolded words or action verbs like explain are going to indicate points. Okay, so here we have a point in part A, and this is going to be really how we earn our points, our money, if you could think of it that way in our FRQ. So explain how the deletion of a gene can change the fitness of an organism. Now I am going to also underline the term fitness because that is a biological term that we've learned throughout the year, and I want to make sure that I include that context in my answer. So how does the deletion of a gene change the fitness of an organism? Now, I know that fitness in biology also basically means the ability to reproduce, okay, based off of Darwinian evolution. So if we're looking at this, explain how the deletion of a gene can change the ability of an organism to reproduce. Well, a lot of being able to reproduce is being able to survive to reproduce. Okay, so if a gene is deleted, that is essential to an organism's survival. Losing it may not allow the organism to survive, to reproduce. Without reproduction, the organism's genes won't be passed on to the next generation. Okay, and will thus lower its fitness. So essentially, we're just answering if a gene is gone and it doesn't allow the organism to survive, the organism can't pass on its genes, and therefore its fitness goes down. Now, there's probably way simpler ways that I could have phrased this, but this just sort of came to mind, but it touches all the bases. And the really key thing I want to point out here is that you don't have to be an expert geneticist or really have memorized the entirety of the genetics unit to be able to understand this, or the evolution unit. It's just simply understanding that if an organism can't, survive or function properly because the gene is gone, then it's not going to be able to reproduce, and if it can't reproduce, it's not fit. I mean, it's just very simple logic. So we've gone ahead and explained. So part B, let's go ahead and take a look here. So we have identify in the word and. Okay, so when we pair a word like and with identify, it means that there's 2 points involved here. 1 point in the identification of the dependent variable and 1 point in the identification of the independent variable. So the and adds on a point to this word identify, making it a 2-pointer. So identify the dependent variable and the independent variable. Oh man, if only we had done this earlier. Now here's the thing about answering AP questions. I wish I could just go ahead and take this and copy and paste it right in and just be like, haha, got you this time, AP. There you go. That's your answer. I'm done, right? No. AP does require you to write in quote-unquote complete sentences. Now what this means is that there needs to be a capital letter at the front and a lowercase letter at the back. So with a few small changes, the independent variable, okay, is the genes the researchers deleted or left as wild type. Okay, so I just elaborated just a very bit more and turned that into a sentence. And then I can go ahead and say the dependent variable, which I don't really think they would care if you left them capitalized, but whatever. The dependent variable is the insect's resistance to insecticide. I want you to be careful here. It's true that it is resistance to insecticide, but when we look at how it's defined in terms of the graph, it's the percent of insects that survive. Okay, so we can say here portrayed or measured as the percent survival of insects. Okay, I'm just making sure to add that so that I can cover all my bases. So we've gone ahead and gotten those 2 points. Very easy because we already did it before. And now we just need to justify the need for each strain to be genetically identical except for the deleted genes. So they're asking us to justify the control. Okay, so essentially the control is supposed to be a measure of comparison with your experimental group. It allows you to be able to see whether or not there is a significant difference in the results of the experimental group versus your group where you kind of keep everything the same. Okay, so in justifying, which I'll go ahead and underline because it's a point, in justifying the need for each strain to be genetically identical except for deleted genes, we're basically trying to justify our control. Okay, we can go ahead and say if the strains weren't all genetically, or actually, you know what? I'm just going to go ahead and explain it this way. Making all strains genetically identical ensures that no extraneous, or that can also mean outside variables, such as fitness or other genes can affect or bias the results of the study to show how each gene affects insecticide resistance. So here I'm literally just coming up with this off the top of my head as you can probably tell. But basically the idea is that we just need to explain that if we didn't have all of the genes or all of the strains genetically identical, that means that genes outside of the ones that we're looking at could have influenced the results of the study and made it seem as if our results were more or less significant than they actually were. So essentially all it's doing is to try to control for any outliers or bias that could be making it into the experiment. And that's really what they're asking you. Why is it that we need this control? Why is it that we ever need a control in a scientific experiment? So by making all strains genetically identical, we make sure that no outside variables such as fitness or other genes could affect or bias the results of the study to show how each gene affects insecticide resistance. So I hope that makes sense to you. We've essentially just justified the need for a control. And part C, using the means and confidence intervals in figure 1, we're going to justify the claim that ABC8 is effective at providing resistance to beta-syfletrin. Okay, so using the means and confidence intervals. Okay, now when I talked to you about this before, we've already gone over this, the means and confidence intervals are going to show us whether or not results are significant. And we said that ABC8 is the most important gene to insecticide resistance. We can also see that if we go ahead and compare the range of its error bar, it's far below the range of the error bar for when all of them are normal or wild type, and even far below the possible highest range of when we just delete P450. So ABC8 causes the largest drop between the two. Okay, so here we're going to go ahead and say the deletion of ABC8 causes the largest drop in percent survival of insects, and we're going to go ahead and say the largest significant drop in percent survival of insects, going from, and I'm going to use the actual data here, I'm going to cite it, going from around 70, a mean, and now when I say mean, I'm going to be using this right here because this is obviously the mean. So I'm going to use this, it's around 70. So going from approximately 70% to, and let's see what it drops down to here. And I'm going to use the mean again. So if I'm using the mean, it drops down to about 20. Okay. So to approximately 20% with no overlap in the error bars, indicating, and actually we can go ahead and say that because we already said significant. So the deletion of ABC8 causes the largest significant drop in percent survival of insects. The deletion of ABC8, and we should probably say alone, because we're making sure that we're saying specifically when we delete this one, causes the largest significant drop in the percent survival of insects, going from approximately 70% to 20% with no overlap in the error bars. So we've gone ahead and used the confidence intervals, which are represented by the error bars, and the means. We've specifically stated the means, which is very good. You always want to use that specific data when you can to justify the claim. Okay, therefore, ABC8 must be vital for the for providing insecticide resistance. Okay, and actually there's another way that we could justify this as well, actually. So this sort of gives us one piece of it, but the other piece that we could use to justify this as well is that if we look at P450, or if we look at, you know, the deletion of just CPS alone, neither of them are as low as just when the deletion of ABC8. So we could go ahead and say neither the deletion of P450 or CPS alone, we'll go ahead and say provides or causes such a significant drop in percent survival, and I'm sort of being lazy here with my typing, percent survival with CPS not being significantly different than when all genes are wild type. And that just sort of allows us to be able to compare all of them. So those are a couple of different ways that you could do it and compare this. But essentially what we're just trying to do is use the actual means in the data and the confidence intervals to make sure that we're explaining why it seems as if ABCA8 is the most important gene involved in insecticide resistance. So hopefully that makes sense. We have justified this claim. We'll move on to part D. P450 encodes an enzyme that detoxifies insecticides. Now here we're back to a lot of contextual information. I'm going to go ahead and start underlining things. So P450 is going to detoxify insecticides. What does detoxify mean? I'm going to go ahead and assume that means something like disable. So it's going to make those insecticides ineffective. ABC8 is going to encode a transporter protein that pumps insecticides out of cells. So at P450 detoxifies and ABC8 pumps it out of cells. CPS encodes an external structural protein located in the exoskeleton that greatly reduces the absorption of insecticides. Now notice how I skipped a lot of the words in there to get directly to absorption, reduces the absorption of insecticides, because I want to know specifically what does it do. Okay, not how does it achieve it, what does it do? Okay, P450 detoxifies or disables insecticides. ABC8 pumps them out of the cells. CPS reduces the absorption of insecticides. That's all I need to know. Okay, so based on this information in the data in figure 1, make predictions. All right, here we go. Make predictions. And notice the S on the end of predictions, because we're making more than one, that explain how a deletion of both P450 and ABC8 result in lower survival of bedbugs compared with the deletion of CPS only. Okay, so I'm going to go ahead and underline deletion of both P450 and ABC8 compared to deletion of CPS only. And then we're going to justify. So let's go ahead and make some predictions here before we even get to that justification part. Okay, so what's going to happen when we delete P450 and ABC8? Well, let's take a look at what they do. P450 disables the insecticide. Okay, that's useful. ABC8 is going to pump it out of the cells. Okay, so if only ABC8 was damaged, then the insecticide wouldn't be able to be pumped out of the cells and it would be trapped inside. But if P450 was still alive, the cell could just detoxify the insecticide even though it was still inside, so it's fine. If P450 was gone, so we couldn't detoxify the insecticide, but ABC8 was fine, ABC8 would still be able to pump the insecticide out of the cell, and the cell would be free of it, and it wouldn't matter. If CPS was the only one that was remaining, okay, it could reduce the absorption of insecticides, but notice the word reduce. It would still let some in, and if P450 wasn't working, it wouldn't be able to detoxify it. ABC8 wouldn't be able to pump it out, and so we'd be slowly leaking an insecticide that would eventually kill the cell. So how would the deletion of both P450 and ABC8 result in lower survival of bedbugs compared with the deletion of CPS only? Well, if P450 was disabled, the cell would be unable to detoxify the poison, and if ABC8 was disabled, the cell wouldn't be able to pump the poison out. Either gene, well, okay, so let's go, okay, so we've described that, okay, cell wouldn't be able to pump the poison out. If both of these, now, let's say, if only if only CPS was disabled, then the cell would still be able to detoxify and pump out the poison. But if P450 and if only P450 and ABC8 were disabled, the cell would still let in toxin slowly and would eventually die. Okay, so essentially we could probably get rid of this first sentence here because that's what they've already given us. So if only CPS was disabled, the cell would still be able to detoxify and pump out the poison using P450 and ABC8. If only P450 and ABC8 were disabled, the cell would still let in toxin slowly and would eventually die. Okay, so we've gone ahead and given our prediction just based off the descriptions they've given us here. I didn't even go back to the graph or any of that stuff to look at it. It's all given to me right here. Okay, so then the last part, justify those predictions. So justify the predictions with reasoning. Okay, they didn't say with data, with reasoning. Okay, so in justifying those predictions, I may have already gone ahead and done that because I basically added this as part of that prediction. Okay, so well, let's go ahead and see if we can add on to that to better justify it. Okay, so CPS only reduces the amount of toxin that enters the cell. It doesn't completely stop it from entering. As long as some toxin can enter the cell will die, especially if it can't detoxify or pump out the poison. Thus, P450 and ABC8 are more important in keeping the cell alive than CPS, as they can deal directly with the poison, whereas CPS is only involved in helping to regulate its entry to the cell. Okay, so I've gone ahead and I provided a little bit more context and a little bit more explanation for this one. Now you can probably do it in far fewer words than I did. Again, this is just off the top of my head. But essentially, I'm just using my reasoning and my logic and what they've given me in Part D to answer this question. I didn't have to go back to Part B or Part C or Part A or the graph or the introduction. All I had to do was just use the logic that they've given me. It's identifying the pieces of relevant information, just the small chunks that you need. That's really going to help you to be able to answer these FRQs. Doing that is just asking yourself the question, what does P450 do? Not how it does it, but what does it do? Detoxifies insecticides. Okay, I'm looking for verbs. Pumps, detoxifies, reduces. Okay, and in that way, I'm able to pull out the important information. I've gone ahead and justified my claim. I've made predictions, and there we go. So I hope this helps you guys. The key things I really want you guys to go back to is go ahead and underline relevant information such as names, verbs, descriptions of experimental controls or variables. Go ahead and come up with a scientific question in the very beginning so you can identify what it is they're asking. Make sure to identify their variables, give them a name and their controls. Go through, read the graph, describe the relationships. If it's not something like this, if it's a relationship with the scatter plot, ask yourself, as one thing goes up, what happens to the other thing? As one thing goes down, what happens to the other thing? Okay, so ask yourself that relationship to those variables and write it down so that you understand it. And then as you go through the questions, make sure that you're underlying or paying attention to the bolded words, how many points that you're getting, and any other keywords that might be there that they require you to use, such as justify using means and confidence intervals, okay, or using reasoning. Then go ahead and provide the most specific response that you can without being as wordy as you can. And of course, obviously here I could have been a lot better if I had had more time to edit. Now, another thing is if you go ahead and use some direct things from the data, this can be a great way to strengthen your response as you're showing them that you're able to read the graph, understand what it's telling you, and to be able to identify the important and necessary parts of it. And finally, for Part D, just make sure that you read through, underline that important information, and then when you're giving your answer, make sure that you're paying attention to all the details and things that they want you to include. If you really look at this, I didn't use a lot of content knowledge for this. I used what they gave me, okay? Now, you will still need content knowledge, it's true. But come in knowing that they're going to give you a lot of, if not most of, what you need to solve these problems. All you need to do is have the simple skills of being able to identify variables, read graphs, and just basically sit back, distance yourself from it a little bit, and identify those important and key scientific things. Scientific question, variable, all that sort of stuff. That's what you want to answer these questions. I know I've repeated myself a bunch, and I know this is a very inefficient video, but I really hope it helps. Guys, you're going to do awesome on the exam. You guys are absolutely amazing, and I count myself very thankful to have been able to be your teacher this year. I look forward to hearing back from you guys about the exam, and I look forward to closing out the rest of the year with you guys, doing fun stuff about biology, and just enjoying this really incredible world we live in. Stay safe, stay healthy, and good luck.